I'm going to talk about structure variation and structure variation discovery. And I would like to also thank you for coming to my talk in such a beautiful day. So I broke my talk into three main groups. First, I will talk about what is structural variation and why we are interested in finding structural variation in the context of human genome. Then I will talk about the methods that have been developed for discovery of structural variation, specifically what are the main signatures that are used for finding structural variations, and what are the tools that have been developed around those signatures. I won't go too much into the details of the method, but I will try to convey the high-level ideas of those methods. And then I will talk a little bit about the results that has been found by applying those methods into whole genome sequence data for different biological data sets. So starting from the basics, when we compare different genomes with each other, for instance, two different human genomes, we might observe different type of variations. And historically, these type of variations have been broken into three categories. One is, of course, the simplest one is CNVs, is that one base pair changes to another base pair. Then it's indels, which is defined as small insertion or deletions. And here, small is kind of a, historically is defined somewhere between 50 base pair or lower. So any insertion or deletion less than 50 base pair, it's called indels. And structural variations are other type of rearrangements, large rearrangements, which change the context of the genome versus another genome. And this can include large CNVs, specifically larger than 50 base pair. So structural variations can be grouped into different classes. And here is a set of structural variations, but these are just simple structural variations. There are more complex structural variations that you can think of. And these simple structural variations can be grouped into two main categories. One is copy number variations. And copy number variations are structural variations that change the number of specific segments copy in the genome. For instance, a deletion deletes a segment of the genome. For instance, it's deleting this segment of reference genome in comparison to the donor genome. This segment is deleted and the copy of that region has reduced. That's why we call it copy number variation. Or a segment is inserted, increases the copy, or a mobile element or retrotransposition is inserted, increases the copy of this region, or we might have tandem duplications or interspersed duplications. So all these set of copy structure variations are categorized as copy number variation. Another group is balanced rearrangements, and these includes things like inversion or translocation. Here, the number of the copies of the segment does not change, but the orientation or location of the segment will change. For instance, in inversion, a segment of genome is inverted, or in translocation, a segment is cut and pasted to another location. It's important to note that there are a lot of diseases that we can find structural variation being causative variation for them. For instance, for autism or intellectual disability, there is a set of copy number variations, which is known that those set of copy number variations can cause with high probability autism. Or for instance, for mobile element, it can cause blood disorders, or schizophrenia, it can be caused by duplication. There are duplications known that can cause this disease. Or for instance, for translocation, famous example is a Philadelphia chromosome, which a translocation of the region of genome can cause a gene fusion, which can result into a specific type of leukemia. So the whole idea in this slide is that one, structural variations can cause a lot of diseases, and two, there's a very different variety of structural variations which we should be looking for. And this list is only the popular ones. There's a lot of more rare ones which I have not put in this slide. So then the question is that, what is the mechanism of creating structural variations? People are working a lot on this and finding different mechanisms for structural variations. Some of the more famous ones are listed here. The most famous mechanism for creating structural variation is non-allelic homologous recombination. That's when during recombination, two segments of genome which are not non-allelic, but they have homology, for instance, two segmental duplications or two repeat, are recombined by mistake and produce a duplication or deletion. And that's known as NAHR. And majority of deletion and duplications that we find is believed to be caused by this mechanism. Another one is retrotransposition. That's when things like mobile elements copy themselves to another location in genome. Another mechanism for creating structural variation, specifically smaller structural variation in non-homologous end joining, that's when during errors in the genome, when the non-homologous end joining mechanism does not work properly and does not fix the error. And there are more advanced mechanisms, for instance, FASTs, which work by template switching, and they're believed to cause complex structural variations. So now that we know a little bit about the basics of structural variation, how can we find structural variations? First, it's note that we know that the frequency of the variation is dependent to the size of variation. In other words, the most frequent variations are the smallest ones, single nucleotide changes, then the copy number changes, and the larger ones becomes very, very rare. So traditionally, the way to assay all these variations was done if you were looking for SNPs, only for exome and CNVs, SNP genotyping was the best way to do. Still it is, it's very high throughput, but only can target a very specific type of variation. We can use array CGH for finding CNVs, which is the throughput is good, and it can find CNVs above some specific range. For very large CNVs, carrier trapping was used, and next generation sequencing, which is the part that I will be focusing on, a lot, can cover all type of variations. So that's the power of next generation sequencing, because it gives us the capability to not only cover every range of variation, but also is quite high throughput and quite cheap. So we know that with the advent of high-throughput sequencing, genomics has been revolutionized. Now we can sequence a lot of genomes very, very fast and very cheaply. You have all seen this slide, which shows the cost of sequencing dropping significantly in the past few years. Now we can sequence a whole genome. A human genome can be sequenced with a cost of around $1,000 if you're a good negotiator, and that's amazing. That means that we can get our genome sequenced with almost the cost of a laptop. And this has given us the capability to sequence thousands and thousands of genomes. For instance, just last week, the 10,000 whole genome sequence with high coverage was released by a company, which that was not imaginable 10 years ago. But this extreme power of whole genome sequencing, and especially high-throughput sequencing, has its drawbacks too. Mainly, the reads that are produced by these technologies tend to be quite small. For instance, the most common type of technology, Illumina, produce reads between 75 base pair to 200 base pair. And it's important to note that the human genome is riddled with repeat. There are repetitions all over human genome. It's believed that around 45% of the human genome consists of these repeat elements. This includes segmental duplication, mobile elements, and so on and so forth. So combination of having a short reads and huge amount of repeat elements makes variation discovery, especially structural variation, a tough task. So a little bit detail about how paired end sequencing and whole genome sequencing works. First, the genome, the donor genome that we are interested in to sequence, is fragmented into small pieces. This small fragment is given to a sequencer, where sequencer sequences the two ends of the fragment for each of these fragments, producing what's known as paired end sequencing. And the two ends, the length of each end is somewhere between 75% to 200 base pair based on technology, and now it's growing. But it's important to note that the distance between the two ends, which is equivalent to the length of the fragment, usually obeys a normal distribution, which we have lower bound and upper bound in the size of this distribution. In other words, we know what the length should be. We have an approximation distribution of this length. And this will be very helpful for calling structural variation down the road. So what are the main signatures that we will be using for calling structural variation? There are four main signatures. One of them is read pair analysis, and read pair analysis uses that distribution that I just showed, and it looks at the reads, and when the paired end read is mapped back to reference genome based on the difference from the distance of the two ends mapped versus what was expected, tries to call structural variation. In other words, if the distance between two ends is much larger than what you would have expected, that indicates a potential deletion in the donor genome. So this read pair analysis is typically used for calling deletions, small insertions, inversions, transposition, and we will see how that's done. And it has a pretty good breakpoint resolution. It can't exactly tell you the breakpoint, but with a high enough coverage can get pretty close to the breakpoint of the events. The second signature that's quite commonly used for calling CNVs, specifically deletion and duplications, is read depth analysis. And the idea over there is that if there is a copy number happening at a particular location, the number of reads that are mapping to that location should change. In other words, if we have a deletion, the number of reads mapping there should get reduced because now we have less copy of that particular segment in the genome that's been sequenced. Or if there is a duplication, that we should see an increase in the read depth of that location. So that's the idea behind read depth analysis. Split read analysis is that when you map the read, if the read gets broken into subparts, that's the indication of a potential rearrangement. For instance, if we have a read and it gets broken into 2 segments as such, probably there is a deletion happening here in the donor genome. In other words, there's an extra segment here in the reference genome. That's one explanation. There might be other explanation for a read breaking into subparts. And the good thing about this analysis is that although it's expensive, but it can give you base per resolution, 1 base per resolution for finding variations. And finally, something that's becoming recently popular is to use local assembly or global assembly to call structural variation. In other words, we can get the reads that are mapping to a particular location, run a particular assembler on those reads, create a larger contig, and then align the 2 contigs together and call variation or structural variation. So these are the 4 main signatures, and we will see how these 4 signatures methods have been developed for each of them for calling structural variations. But before continuing, let's ask the question of what is the main challenges of calling structural variation. One of the most important challenge for calling structural variation that to a good extent is being ignored is that structural variations are not distributed uniformly throughout the genome. Because of the mechanisms that create structural variation, as I showed, most of them are homology-driven. Structural variations tend to cluster near repeat regions of the genome or segmental duplication. And the fact that those regions are enriched in structural variation makes structural variation calling harder. So if we ignore the repeat regions and only look at the regions with the unique mapping, we will lose sensitivity. If we look at the regions with repeat mapping, with duplication, which have multiple mapping, we have a potential of having higher false discovery rate. And another important thing is that reference genome is not complete. Although human reference genome, we have been working for a very long time to make it a complete clean reference, still there are regions of the genome which is missing, and that can cause a lot of problem. And the other thing is that the reference genome regions in it can be completely misassembled. There are regions that the assembly in human genome is completely wrong. And those are the 2 points to have to be careful when we call structural variation, especially in those regions. So for instance, a lot of the tools have black regions that they don't call structural variation in those regions because they know the false discovery rate will be really high for those regions. So finally, a lot of structural variations can become complex. By this, I mean that they are not just a clean structural variation with 2 breakpoints. They might have different variations near breakpoints, or they might have multiple structural variations overlapping with each other, which can cause complexity. So let's look at the signatures that I talked a little bit in more detail. The first one was read depth signature, and that's when the number of the reads mapping to a particular region significantly is different than what you would expect. So the assumption of most of the methods that use read depth signature for calling CNV is that the read depth distribution should obey a Poisson distribution. And based on that assumption, as a matter of fact, the very first methods for calling CNVs were using Sanger reads, and they were using read depth analysis. One of them was called WSSD, and it used Sanger sequencing and Megablast for mapping. So you can see how old the first method for calling copy number variation were. So there are now new methods which are developed for NGS. They, for instance, they have higher resolution like CNVinator and a few other tools. So I would go a little bit into how this whole old WSSD works. Is that it of course has the reference genome, it has the reads, maps all the reads to the reference genome, and then looks at the windows of 5 kilobase per and calculates the read count for each of those 5 kilobase per windows and shifts it by 1 kilobase per. And for every region, it tries to predict if there is a deletion, if there is a unique region, meaning copy number 2, or there's a duplication based on the reads that it observed. So that's the whole idea between read depth analysis and WSSD. But there are a couple of the things to be careful here. One is that do we use multiple mapping or unique mapping? Assuming that this is the genome that we are sequencing, and let's say there are regions with duplication, for instance, segmental duplication, if you use multiple mapping and use the read depth analysis, for those regions you will call a higher copy, exactly because they do have higher copy for reference genome. But there might not be different between donor genome and reference genome. So if you use unique mapping, you might call this copy number 1 because you put each read to only one of the locations. So you get higher possibility of finding new copy numbers. So the question here becomes, what is the application? And based on that, you can decide what type of mapping to use. So of course, WSSD was extended for using next generation sequencing technologies. For that, they had to develop, we developed a new mapper called Mr. Fast and Mrs. Fast, and that's designed for exactly mapping each read to multiple locations in a very fast fashion. And then you can apply the same principles with one small caveat. And that's the next generation sequencing has a huge bias for GC, which means that there are regions of the genome with the GC which are high in GC or low in GC, which can cause differences in the coverage, and that needs to be fixed. So one of the other methods that, as I said, was developed for calling CNVs using read depth or CNVinator, it uses a unique mapping of the reads, filters the reads with low quality, and then uses image processing algorithms for finding regions in the genome which have a deviation, which have a change in the read depth. So the GC problem that I mentioned, or the GC normalization that we have to take care of, is that the read depth is highly correlated with the GC of the contact that is being sequenced. So this shows the experiment done on Illumina data for the same segment sequence in different conditions. X-axis is the GC contact of the windows of 50 base pair, and y-axis is the relative read depth. As you can see, for the regions that are very low GC or very high GC, the read depth drops significantly. And we have to normalize that and fix that. The way that most methods use, and the simplest normalization that you can think about, is that for every GC, calculate how much for that particular experiment the read depth is different from expected, and use that factor for normalizing all the read depth values. And by this simple normalization, in most methods, it's performing quite well for fixing the GC issue. So most of the methods that I present for read depth analysis, and from now I'm going to present, are going to be for whole genome sequencing. But I would like just to mention whole exome sequencing for this one slide. Whole exome sequencing is becoming very popular, especially for medical analysis. And what happens is that only the exonic regions of the genome are captured and sequenced. And that consists only 1% of the total genome, and that makes the cost of sequencing the exonic region much, much cheaper. So it's very, very profitable for specific applications. And read depth analysis is used for calling copy number variation for using whole exome sequencing. The only caveat here is that the capture efficiency for every exon is very different. So the assumption of the Poisson distribution is not correct here. So what most tools do for fixing this issue is first they look at many samples that have been whole exome sequence, learn the pattern of efficiency of capturing for each exon, remove that or fix for that, and then do the copy number variation. There are a couple of methods, there are many methods, as a matter of fact, developed for copy number discovery using whole exome sequencing. The two that I'm familiar are XHMM and Conifer, which work quite well for calling copy numbers on large regions of the genome. So the next signature that I will be talking about is read per signature, and that's the signature that uses the information that the length between two ends of the pair then should obey a normal distribution, and the orientation should be a specific orientation. So this is the distribution of the two ends that I was talking about, and it should be between delta min and delta max. And when the reads are mapped back to reference genome, two things can happen. One is that the reads that map exactly obey what we were expecting, and those reads are known as concordant, the black ones here. The distance is as you expected, and the orientation is correct. But sometimes the reads that have been mapped, they don't obey what we expect. For instance, in this example, these red reads, they are mapping farther from each other. And this is known as discordant reads, and that indicates a potential structural variation. So let's look a little bit more in detail how this looks like. Let's assume this is the donor genome, and this is the reference genome. And let's say these are the structural variation. We have an insertion in the donor genome, we have a deletion in the donor genome, and we have an inversion. So if a paired end read spans the insertion, When it's mapped back, the two ends is going to map closer. If it maps if it spans the deletion, the two ends will map further. And if it spans the breakpoint of an inversion, the orientation of one end will change. So these are the signatures in general that's going to be used for calling structural variation using read-pair analysis. So this is the laundry list of different structural variation and how read-pair analysis is going to be used. This is the deletion as I talked about. Mobile element insertion, you're going to have one end mapping to the genome, the other end completely mapping discordantly to a mobile element, and that's how you know a mobile element insertion has happened. You might have inversions, as I said, you might have two different breakpoints and two different type of signatures here to call inversions. Even you can use read-pair signatures for calling duplications, interspersed duplication and tandem duplication, although almost very few methods use this signal for calling duplications. So just one more time, the distribution that I was talking about, the reads that the pairs that map outside of this distribution are the discordant ones and the ones that we are interested in. So there are a lot of methods developed for calling structural variations using read-pair. Maybe 30, 40 methods till now have been developed for using read-pair signatures for calling structural variation. Some of them use unique mapping. I put some of them like breakdancer, genome strip, GA3 here. Some of them use multiple mapping, variation under common law model, Hydra, GS3A Pro. And some of them use the advantage of using multiple genomes together for calling structural variation. So what they do is that instead of calling structural variation in one genome against reference genome, they look at many genomes together. That increases their power significantly in picking up structural variation, which is quite amazing. And there are quite a few tools recently developed for exactly doing that. So almost all of the structural variation algorithms using read-pair have two steps that they perform. In the first step, they try to find a set of reads, set of mappings that they belong together and they support a particular structural variation. And they have different terminologies for it: maximal value clusters, maximal intersecting break-unique, maximal cliques. So there's a maximal set of reads that have similar property and they support the same structural variation. They find these maximal clusters, and then using different objective functions, they try to pick some set of these clusters as their predicted structural variation. So here is 3 different examples of this maximal stuff reads that I was talking about. One of them is maximal value clusters, and I'm showing these all 3 in the context of deletion. Let's assume that there is a deletion happening between breakpoint left and breakpoint right in the donor genome. If you have 2 paired end reads, if these 2 paired end reads want to be part of this maximal cluster, they should obey a simple logical rule, and the logical rule is that the distance between breakpoint left and the left end mapping of the read, and from the right breakpoint to the right mapping of the in for both of them should be between delta min and delta max, because that's the distance of the paired end read. Or you can model it in a geometric way, as was done in Cindy et al. You can consider every breakpoint, because you have 2 breakpoints for deletion as a 2-dimensional space. You have one genomic coordinate as x-axis, the other genomic coordinate as y-axis, and every paired end mapping is a trapezoid in this space. Now, if you have multiple paired end mapping that want to support the same deletion, they should have an intersection at a particular location. This location is a breakpoint that supports the deletion. Very similar to here, that this breakpoint between breakpoint and breakpoint right, you needed to find a breakpoint that supports this logical rule. Another way to look at it is using a graph. That's what was done in Marshall et al. You can consider every read that you have as a node in this graph, and if they are supporting the same structural variation, you can have an edge here, and now try to find the maximal cliques. So you find a maximal value cluster, or maximal intersection breakpoint, or maximal clique. The idea is to find a maximal set of reads that are supporting a particular variation. So this was the case of deletion. You can apply the same concept on the case of, for instance, mobile element insertion. You have a mobile element insertion, let's say ALU, which copies itself to another location in the donor genome, like here. And mobile elements I'm very interested in them, and I think they're quite cool because they have the power to copy themselves to any location in genome. And for instance, ALU element, there is over 1 million copies of ALU element in human genome. 10% of our genome is only this 300 base pair element that just jumps around and still is continuing jumping around. So we are very interested to be able to find these insertions. And of course, again, you can use the read pair signatures if I assume that these are two read pairs that are inside the ALU that has been an inserted ALU. When you map it back to a reference genome, they're going to map discordantly. And again, you can use the same concept of finding maximal set of reads that support this insertion. We showed that we can find this in an order n log n, which is quite great, and we can find these maximal clusters. Then, if your reads that were considered were mapped only uniquely, you have these maximal clusters, you can come up with some statistical model or some scores and pick a set of those clusters as your structural variations. The problem becomes slightly more challenging if you have multiple mapping reads. In other words, if one read has mapped to multiple location, now that read can belong to multiple clusters or multiple clicks or multiple breakpoint regions that you're considering. And then you can have two different objective functions that have become popular. One is that you can say, I want a maximal parsimony objective function. In other words, I want to find the minimum number of structural variation which covers my universe. Or you can have some sort of probabilistic formulation, for instance, maximum likelihood estimation. And there are quite a few methods developed for trying to solve this problem. So I'm going to just go into one of them for maximum parsimony structural variation. So the goal over there is to assign each read to only one location, such that the total number of structural variation is minimized. And it's been shown that that problem is NP complete using reduction from set cover problem. And one method for solving it is to first find this maximal valid clusters, as I mentioned, and then pick the minimum number of maximal valid clusters to cover the universe, and that gives you an order log n approximation similar to what was happening in set cover algorithm. So that was all about read pair signature, but there is another signature which is becoming quite popular and very powerful, and that's split read signature. Split read signature is that when a read, when it's mapped back to reference genome, breaks. In other words, the read cannot be mapped completely. Some parts of it map to one location, other parts map to another location. So how does split read help, for instance, for calling deletion? As you can see, if the read breaks as follows, probably it's a deletion. Or if it's a mobile element insertion, it's going to break one part here, the other part will go to a mobile element in the genome. If it's an inversion, you will see a very interesting signature is that one end maps perfectly, the other end breaks, one end maps in correct orientation, the other one changes orientation, and so on. So at the beginning, it took some time for the methods to start using split read, mainly because the read length was quite small. For instance, 36 base pair, and thinking about breaking 36 base pair was quite hard. But still, there were methods developed like Pindel, which exactly tried to do that, and they were very successful in calling small indels. But now that the read lengths have become longer, we have 100 base pair, 150 base pair, using an approach such as split read is very exciting, and we can use split read to call structural variations. And quite a few methods have been developed. For unique mapping, I'm just mentioning a couple here, but there are a lot of other methods have been developed for exactly doing this. Even BWMEM tries to now break the read to a soft clipping or hard clipping and gives you that information which you can use for calling variation, including indel structural variation. And there are some which are explicitly designed for calling structural variation in the repeat regions of the genome using split read approach. One of them is called split read, but it works quite well, especially for whole exome sequencing approaches when you have a non-uniform distribution of reads. So I'm going to only show you the details of one of the methods. Pindel, as I said, that was the first method that introduced using split read signature for calling structural variation, and it works as follows. Because breaking the reads can be quite expensive, the trick that they use is that first they only consider reads that one end, because we have paired an information, one end has mapped perfectly and the other end has not mapped. So they say only let's look at those minority of the reads that have such a property, and then let's break that read that had such a property such that one end is close to the one part of the read broken is close to the read that has been mapped, and the other read is some distance away. So based on this criteria, they can make Pindel much faster than if you would have just tried to break it or align a read by breaking it to any part in the genome. So finally, Another type of analysis that's been used for calling structural variation is using assembly. And based on assembly, it's easy. You try to get the regions. Well, in theory, it's easy, but in application, it becomes very hard. Is that you try to get the reads that are mapping to a particular location, give it to an assembler, treat an assembler as a black box, give it to the assembler, get the contig, and then align the contig back to a reference genome, and then call structural variations. For instance, if you have a deletion, you will perfectly see a contig which that segment is missing. Or if you have an insertion, you have an extra region. Or if you have an inversion in your contig, the whole segment will be inverted. So there are two ways to use this assembly approach. One is to do a global assembly analysis. You just get all the reads that are a whole genome sequence, give it to an assembler, get all the contigs that you get and align those contigs back to the reference genome and call the variations. Another way is to use some sort of local assembly analysis. Let's say you're interested in a specific region. What you will do is that you will get the reads that are mapping to that particular region, give it to an assembler, build a contig, and map the contig back or align the contig back. So you can focus on a specific region that you're interested in. This increases the accuracy and speed of calling variations if you know the region or you just want to validate a particular region. And there are a lot of popular assembler tools out there. I'm just mentioning a few like Velvet, Euler, Abyss, Cortex, and so on. So I will go into a little bit more detail for two of these tools that use assembly approach for solving structural variation, which are a little bit more specialized. One of them is NovelSeq, and this tool was developed specifically for finding novel sequences inserted in one genome versus the other one. And by novel sequences, we mean that the sequences that do not exist anywhere else in the genome. In other words, brand new sequences inserted. And the idea over there is that if you have the paired end reads and map the paired end reads back to a reference genome, if there are a set of reads that do not map anywhere, they potentially can belong to this new contig, new sequence that has been inserted in the genome. So we can get those reads, which they call orphan, give it to assembler, build these orphan contigs, and also get the reads which only one end is mapping and the other end is not mapping and they're cluster, and also assemble the ends, and then use by-part matching of matching these one end contigs with the orphan contigs, and potentially predict novel sequence insertion. And in practice, it was capable of finding new novel insertions specific for some specific population as part of the 1000 genome project. Another assembly approach which I really like and I think it's really cute is that it uses the Debrien graph approach or the assembly graph approach for variant calling before building an assembler. So what it does is that let's assume that you're interested in calling variants from one genome against the reference genome. Instead of mapping the reads back to the reference genome, how about we bring the reference genome into the Debrien graph world? In other words, let's assume every k-mer that we have is a node in the graph, and we have colors based on if it's coming from the new genome assembled or it's shared with reference genome. And then if there is a paths in this graph which are unique color, those paths indicates potential variant. Because if it's not a variant, both genomes should have those k-mers or should be paths that are going through them. The good thing about this approach is that you can use it for comparing multiple genomes together. Let's say you have 3 genomes or 4 genomes and you want to know what are the variations between them. You can color every of those genomes separately with different colors and then look at the graphs that have been built. And if you see unicolor paths, you know that there is a variant in that particular sample which is missing in other samples. So this was published, the tool is called Cortex, and as a matter of fact works quite well for calling some specific types of structural variations. So till now, I talked mostly about methods that have been developed to address one type of signatures. But now we can imagine methods that use multiple signatures to improve the results, and that's exactly what's being done. There are quite a few methods that have been done which do that. I'm just reporting some of them here. For instance, DELI, which mostly is designed for calling deletion, uses a combination of split read and read pair signatures together for calling deletions. One tool that recently came out and works quite well in my experience is Lumpy, which uses split read pair, and read depth together. The other one is GSV Pro, which uses read pair and read depth together and has a MCMC formulation for assigning reads to a correct location because it considers multiple mapping. Or Genome Street, which works very well for finding large CNVs when you have many samples sequenced. So it takes the advantage of having many samples, compares them together, especially compares the read depth signatures together to find if there are outliers in those samples and predict copy number variation, specifically designed for calling CNVs when you have a lot of samples, hundreds or thousands of samples. So, linkage analysis. So what they do is that they have also, they also look at the SNP information, also other structure variation. And if they see, for instance, in one sample, there is a high linkage between one structure variation and another structure variation. In another sample, they have one structure variation, the other one, the support is very low. They use the linkage information to say, oh, the structure variation over there is correct too. Between two structure variation and even SNP with the structure variation, so all of them, it's been used here. In that regard, it's quite powerful for especially finding common structure variations. So till now I talked mostly about the methods that have been developed, but how about let's look at some of the results that we can get. So the first result I want to show you is from the 1000 Genome Project, the pilot project, and this shows the distribution of the different types of variations, SNPs, indels, and structure variations. The x-axis is the number of variants observed. This part is the portion that's novel, and these are the length. Of course, the most number of variations observed in 1000 genome are SNPs. It's order of magnitude more than any other variation, but that's not surprising. But as you can see, there's quite a few healthy number of structure variation, insertion, and deletions happening. And if you consider the length here, if you consider the length and the area underneath, the number of the base pairs affected by structure variations is very much comparable to number of base pairs affected by SNPs. So that shows that structure variations are very important in population genomics and they should not be ignored. So how about studying structure variation in Great Ape Evolution or in general in evolution? Is that at one project what we did was, as a large consortium did, was that they sequenced something around 85 Great Ape genomes that includes human, chimpanzees, bonobo, orangutans, and gorillas, and different types of variations were called: SNPs, deletions, duplications, mobile element insertions. And I'm going to show you only the trees that were built, the phylogeny trees that were built for this analysis. This is for SNP. As you can see very nicely, each species is separated. Here is humans, here is gorillas, chimpanzees, bonobos, and orangutans. Works very nicely, and not only that, it also separates the subspecies. Different colors indicate different subspecies of these species. When we do the same thing exactly using mobile element insertion, for instance, allo insertion, we can exactly capture the same structure of species. Human here, chimpanzees, bonobo, gorilla, orangutan. The same for L1 insertion. And this shows that structural variation have a very nice power in informing us about evolution when we're doing evolutionary studies. And another thing that you might notice is that the branch lengths here is not the same between these different types of variations. That exactly indicates that the rate of these events are not the same between each other. For instance, the rate of the SNPs is not the same with the rate of deletion or rate of mobile element insertion. As a matter of fact, although rate of SNPs to a very good extent stays constant during time, at least for a long time, for rate of other structure variation fluctuates significantly during short periods of evolutionary time. So finally, let's look at the structure variation in the context of disease. I'm showing you some examples from the whole genome sequence of the autism, quads, and trios. These are families which mother and father are not diagnosed with autism, but they have one child which is diagnosed with autism. And these results have these samples, these 53 quads and trios were whole genome sequence. And we ran our methods for calling structure variations, and I'm showing only 2 of them here out of this 53. And we found de novo deletion and duplications affecting genes which we believe are important for autism. By de novo, I mean these variations do not exist in the parents, and they are brand new variation only observed in those child. And we believe there is a very high chance that those variations are the causative reason. Another family I'm showing here, this one inherited we inherited deletions from mother and father, and all three deletions that I'm showing here are extremely rare and private to this family. And one of this deletion, CAC-NAT2D4, we were interested because we know this gene is quite important, and it was deleting its exons, so we genotyped this deletion in a large population of cases and controls, and we observed that that particular deletion, which we found using whole genome sequencing and before was missed by ACGH or other technologies, is significantly enriched in cases to control. So we believe studying rare CNVs has given us power to find with a high probability causative events in these children. So this was a study that we did, but parallel to us, another study was happening, which they sequenced 71 autism and quads, and they found a really cool result, and that was they found two de novo structural variation in two different families, which were complex structural variation, which affected genes which are very important. In one family, in one child, they found a de novo inversion, very long inversion, over 1 megabase, which breaks the gene CDHA. And the other one was a complex structural variation, again de novo in that family, which fused two parts of a gene into one segment, which is very strange, and this again happened de novo. Okay, so till now everything looks great. We have methods for calling structural variation. They seem to be working well. We can apply it to the study of population, evolution, and disease. But what are we missing? So recently, one haploid human genome was sequenced using PacBio data, and PacBio data has the capability of producing very long reads. So we don't have the issue with the short read, which a lot of reads we don't know where they map to because these are long reads. As you can see, this is the distribution of the reads lengths that we got from PacBio. They're quite large. So we can really study this genome in depth. And the other thing is that this is a naturally haploid genome in the sense that although it's a human genome, but the two haploids are identical. So we don't have to even worry about heterozygous versus homozygous. Whatever we see, it's going to be homozygous variation. So Mark Tyson in our lab, who developed the main tool for calling reads, aligning reads to the reference genome known as Blazor, which is currently used by every group that uses PacBio, developed a method for calling structural variation, and it works as follows. Looks at all the reads that are mapping to the reference genome, and based on disparity in the reads, for instance, if the reads are broken, a group of them, or there is a place that all of them are ending, it calls them hard stops, gets those reads and gives them to Celera assembler, builds a contig and maps the contig back to the reference genome and calls structural variations. And what we did, he called a lot of structural variation using this approach, and we validated a very large portion of them, and we believe the structural variations we call with a very, very high probability, majority of them are correct. So we sequenced the same sample using Illumina, and we also have the whole genome structural variation called using 1,000 genome, that's 2,500 samples. So our assumption was that majority of the structural variations predicted in this one sample should be covered with BioPSync. But that was not the case. So here, the very reddish thing shows the new insertions which were only seen in PacBio assembled CHM1 versus pink, which was the ones that were shared from what we saw before. And the very black is the novel deletions, and the gray is what we saw and reported before. As you can see based on the figures here, there is a very, very large number of structural variations which we only picked by PacBio, and we did not pick as part of 1,000 genome project or as part of the running a variety of tools for calling structural variation on the CHM1 Illumina data, which means there is still a lot to be learned in this direction. Any questions about this? Because this is quite shocking for a person who works on structural variation that we are still after 5-6 years missing so many structural variations. Okay. So in conclusion, and the future works that our people are currently working, one is that one topic of structural variation which is becoming quite hot and interesting is finding somatic structural variation and copy number variation. In general, finding somatic variation is very hard because the coverage is lower. You might not even sequence the correct cell. It's a very challenging, but it's very important in the context of cancer. It's believed that potentially a lot of the driver variations are somatic CNV or structural variation. And also in the context of neuroscience, there are quite a few papers recently which claim there is a very high rate of somatic mobile element, especially L1, in neurons. So it might happen that a lot of the neurological diseases are because of the somatic variations. It's not known, but we know the somatic rate is really, really high in neurons. And finding these variations is very, very challenging at the moment. Second is that with the third generation sequencing starting to becoming more popular, we need to develop new methods for studying these structural variations. And different technologies have different properties. For instance, PacBio data, Oxford Nanopore, and 10x Genomics, and so on. They each have their own properties, and they need to be developed. We need to develop methods which specifically target those technologies. And finally, we are finding a lot of structural variations using third generation and second generation sequencing technologies, but we are lacking in really storing them in a manner that we can, every new genome that's been sequenced, we can genotype all those structural variations. And we're finding data structures which we can store all these structural variation in a way that any new genome that's been sequenced, we can immediately genotype it and see, oh, which set of structural variations exist there or not. That's something that also is quite challenging, and people are working on it. And I think with that, I finish.